Okay. So a schedule check for us. So we've got this week, and then next week is what? Doesn't say? Oh, President's Day. It is President's Day. After President's Day? Exam. Okay. President's Day is not a university holiday, just so you guys know. So the university is open. In fact, we do have class. You're having an SI in class time, so there's not any new material. Okay? So I fully anticipate large participation on Monday, because I know you don't have any class conflicts, and it's not a university holiday. So it was convenient. It wasn't necessarily scheduled, because I'm actually going to be out of town, but it actually works out really well. I built the schedule around that so that we have the exam on Wednesday instead of Monday. It gave us an extra study day. It gave you an extra study day. So we need to get through adaptive immune system today and Wednesday. That's the last of the material. And I think what you'll find is it'll be a nice sort of package, if you will, of an introduction to pathology. And so today we're going to bridge over from the non-specific immune system to our specific immune system, or our adaptive immune system. And so I want to start out today kind of characterizing the immune system, and we're going to break it out organizationally into a couple of different categories. So when we look at the immune system, we talked about barriers as our first line of defense. You guys remember this? And then we moved into our second line of defense. And this was what? Inflammation. And these two roll up under the immune system. And what category would we have? So this would be the innate or non-specific. You guys with me? And under inflammation, we had a couple of different cell types that we learned about. What were some of those cell types that we learned about? Neutrophils and macrophages. Okay, perfect. So the first responders we had were the neutrophils. And then our second responders were our tissue macrophages. So we got our first line of defense. We have our second line of defense. Under our innate branch of the immune system. And now what we need to introduce is we need to introduce this secondary side, which is called our adaptive or our specific immune system. You guys follow? This is going to be which line of defense now? Adaptive, but going numerically, we'll talk about first, second, and now third. So I want to let you guys in on kind of a little secret in biology or in physiology, in biological systems. So anytime you identify that there's redundancy happening, that should tell you something. What do you think that should signal to you? Anytime you're studying a system, and we borrow this, engineers borrow this from living systems. Engineers, I'm not talking about bioengineers, I'm talking about civil engineers, I'm talking about aerospace engineers. We borrow this concept of redundancy. What do you do when you see something biologically that has redundancy? And then what do you do when you're flying an airplane and you see redundancy? It tells you that that's probably what? Prone to might break? Is it pretty important if you have backup systems? Yeah. So here, you've got 3 systems that are in place in order for us to fight off foreign invaders. So one might come to the conclusion, well, something's going to breach the barriers. It's going to get through. What are some examples of our barriers? This is all review so far. So skin, obviously skin. Hair. What else? Membranes, right? Mucous membranes. Something's going to get past these. Then we've got our second line of defense. We've got our neutrophils and our macrophages. And you guys now understand, hopefully can appreciate that in a wound healing response or anytime there's an invasion or there's damage to tissue, that we want to stimulate inflammation in order to trigger repair. And sometimes it goes down a pathway of significant fibrosis where you got new collagen and new deposition of extracellular matrix, but it doesn't resemble the native tissue or the original tissue. You lose a lot of function, it doesn't look aesthetically pleasing, and it doesn't function like the original tissue. But sometimes it actually goes down the process of complete regeneration where you restore function, you have minimal scar formation. In both of those situations, the inflammation comes up and then it goes back down, right? And you guys would be familiar with those kinds of graphs. In other circumstances, this inflammatory state may actually stay elevated for a long period of time, and we call that what? Chronic inflammation. And that's going to be really kind of foundational in Unit 2 and Unit 3 after this first exam. That's going to be foundational to a lot of the disease states that we're going to go study. The other one is going to be in the adaptive system. This is also going to be foundational. And so that's why Unit 1 is rather critical, because in Unit 1, we learn an awful lot about what's happening with respect to our adaptive or specific immune system. In here, this system actually utilizes specialized cells known as lymphocytes. And there's 2 players. We've got B cells. And then anybody else read ahead? We have T cells. Very good. So we call them B lymphocytes or T lymphocytes. They are not neutrophils and macrophages. These guys had another name. What category were these guys from? Leukocytes. Very good. Now, this kind of sounds like the same thing, but it's not. Spelled differently, right? Another name for a leukocyte is what? A white blood cell. Very good. A lot of nomenclature on the board. So we got our first line of defense, our second line of defense. We're going to talk now about our adaptive or specific immune system, and we're going to introduce new cell types today. Now, if you looked ahead, what did you notice about the length of the slide deck? What's that? It's short. Don't let that confuse you. Complicated things come in short packages sometimes, okay? That's what my wife tells me. You are complicated. I don't think it's a compliment, trust me. So just because the slide deck is short doesn't mean that this isn't challenging material, okay? But it's intentional because there's a lot of information on those slides, and so we're going to kind of slow our roll over the next 2 lectures to make sure that this information sinks in. And that's why I'm taking a little bit of a break from our normal slide deck to kind of go over some of the material, because I want you to have, this is a roadmap, right? What are we doing here? We're drawing a roadmap of the immune system. And we're going to be in the immune system for 2 lectures, 3 hours total, or 2.5 hours total. Somebody like, 3 hours, right? Hour 15 times 2, right? Math wasn't my thing. So 2.5 hours total this week. There are semester-long courses in immunology. We are not going to cover all of immunology in 2 lectures, not in a 2.5 hour period of time. But if I can give you a roadmap so that as you move on in your studies, you're like, okay, man, these T cells, this T cell disease is very complicated. I'm talking about cancer, and it's some sort of T-cell lymphoma, cancer, that they're talking about. Well, now you kind of know where you are. You with me? If it's a leukemia, it's a white blood cell cancer. Where are we? Right here. You guys with me? If we're a barrier cancer, like a melanoma or a squamous cell carcinoma, okay, we're over here. Okay, very different types of cancers, and we're covering cancer in Unit 2, so now you have a framework because you have the roadmap, you kind of know where in the heck are we, okay? So, let's take a breath. We're not going to cover all of immunology in 2 lectures. Some of you have had immunology courses, which is awesome. It'll be very helpful. Okay, if you haven't, and you will, then I think this will be a nice introduction and give you kind of some foundational concepts to help us get through, or help you get through in the future. Now, a couple of concepts about these lymphocytes. You guys know where these guys come from. These guys both originate in the bone marrow, but then they sort of go through a maturation process, or they go through an educational process, and that's really what it is, in a couple of different target tissues. So the B cells are called B cells. Let me see if I can, just want to make sure that this is showing up on the, okay, cool. The B cells, Richard, I really appreciate you erasing this. We may get to it, we may not. But it looks beautiful. The B cells mature in the bone marrow. So they arise in the bone marrow, and they are educated or mature in the bone marrow. So we refer to them as B cells, which is really convenient that bone marrow matches with B cell, right? BB. Any guesses, if you read, where the T cells go through maturation or an educational process? Thymus. Wow, I'm really proud of you guys. That's awesome. This is a very complex lecture, so if you read and you did kind of the homework assignment ahead of time, no one can blame us for going too fast today. So if it feels like you're going too fast and you just kind of got through the pre-quiz but you didn't really do the reading, next time you might try doing kind of what's prescribed, because I think you'll find that it's a lot easier to understand. So this is thymus. Okay, T cells mature in the thymus. So I'm really proud of those of you that have done this. Maybe all of you, 95% of you, or all of you have done the readings. It's great. But today it's going to really pay off. Yes, Max. That's because of where they're maturing. That's correct. The B is bone marrow. The T is thymus. Okay, so it's really convenient. Okay. Anybody know where the thymus is? Yeah, it's about right where my microphones are. Okay, it's above the heart. It kind of in the throat region. Okay. It shrinks as we get older. We'll talk about it here in a second. It's actually the largest in childhood because, well, I can't avoid it. Well, I was going to get to it later, but these T cells become educated in thymus tissue. And because they become educated in thymus tissue, they are learning a tremendous amount. They're at school while you're at school as a kid. And most of the diseases or viruses and bacteria that you come in contact with are at school when you're a kid. And so this thymus actually enlarges during childhood. And then by your 20s and 30s, it's already starting to shrink. And by the time you're elderly, it's almost indistinguishable on autopsy in a 75, 80-year-old patient. You can't really tell. It's kind of gray and all shriveled up. It's hard to find. Okay. So, roadmap. All right, let's talk about the different locations that we're going to find this stuff. So these B cells, they actually manufacture. What do they manufacture? One of you guys tell me what they manufacture. What do they make? Antibodies. Well done. They're going to make antibodies. 5 different types of antibodies that we're going to cover a little bit later. And these T cells, what do? Do they make antibodies? How do they work? B cells act through antibodies, how do T cells act? Cell-to-cell interaction, okay? So it's a cellular response, okay? And many times it's cellular cytotoxicity. So it actually attacks another cell. We'll get to that here in a second. So I think I'm going to pause here and then we'll get to some of the main slides in the deck. And we'll kind of come back to this outline. Okay? A couple more details I think that I'll fill out. T cells. We've got two different flavors of T cells. Two different styles of T cells, right? And these T cells have a label on them. And these labels are what we call a cluster of differentiation. And so on a cell, on the surface, there's going to be some marker. And this cluster of differentiation is labeled CD, and then there's some numerical nomenclature. And so if we've got a CD4, we would say that this cell is a CD4 positive T cell. Okay. We actually could have another cell here, and this cell has a CD marker on it, and it's CD8. We could actually have a third type of cell, and let's pretend that this marker is like CD90 or 105. Well, this CD90 or 105, this is actually a marker, an indicator of a mesenchymal stem cell, like what we learned about in the last lecture. Mesenchymal stem cell. So these labels of CD90 or 105 tell us that that's a mesenchymal stem cell. This CD4 tells us that this is a very specific type of T cell. Anybody know from the readings what kind of T cell this is? It's a helper T cell. Very well done. This is a helper T. And how about the CD8? It's a cytotoxic T. So over here we can fill this out and we can say we could have a CD4 positive T cell or we could have a CD8 positive T cell, and we would know that this is a helper T and this is a cytotoxic. Now you guys can see why I don't write on the board that much, because my handwriting is horrible. Okay? So I'm going to flash forward. I'm going to sort of foreshadow. These CD4 positive T helper cells, they're very important. I mean, they're labeled helper cells. Why do you think they're labeled helper cells? What would you think? They help other cells. Yeah, good guess. You're totally right. Okay? It's like doctor. Nice job. What do you want to call this? I don't know. What does it do? Well, it helps other cells fight infection. Let's call it a helper cell. Sounds good. Okay? That's how that works. So this is a helper T cell. It is really what we call the nomenclature is, this is referred to as the linchpin cell in the adaptive immune system, the linchpin cell. Does everybody know what that terminology means, linchpin cell? Everything kind of pivots on this particular cell, okay, the linchpin. So if it's missing, everything kind of falls apart. So believe it or not, this isn't on this exam, it'll be in the next unit, but the HIV virus eliminates your population of helper T cells. So we'll see that when we study HIV. And by eliminating the helper T cell population, your immune system is rendered helpless. Okay? I mean, the pun intended. And so it is unable to fight off a lot of different types of infections. And so the acquired immunodeficiency syndrome, or AIDS, that's resulting from HIV progressing, you don't necessarily, the patient doesn't expire from HIV. They succumb to some other infection. And they're in a situation where they have an immunodeficiency, acquired immunodeficiency syndrome, because their helper T populations are diminished. So I'm just telling you this anecdote now so you can kind of say, okay, so that makes sense to why, as all of us know that AIDS is fatal, HIV is very dangerous, and now you kind of can connect, okay, the CD4, the activities that we learn about of CD4, in end-stage AIDS patients, they're not able to do that. Okay. So clusters of differentiation, we covered that. We talked about where these lymphocytes come from. We've talked a little bit about what they do. And now let's talk about a little bit of extra detail. So in your body, about 60% of the population of T cells are helper style, and 40% are cytotoxic. So more than half of your lymphocytes, your lymphocytes in your body are T lymphocytes, more than half are helpers versus cytotoxic. Yeah?
What is the MSC that you put down?
Oh, this is from the last lecture. This is mesenchymal stem cell.
Oh, okay.
So all nucleated cells in the body have these labels on them, these clusters of differentiation. So when you're reading in the text and you hear about Cd4 positive, most students would come to me in years past and go, what the heck? I don't even understand what they're talking about. Like, my parents had CDs, but like, we don't use those anymore. Like, what are we? So cluster of differentiation is a label. So I was just giving you an old reference that in a previous lectures, when we talked about wound healing, we looked at mesenchymal stem cells. And so they have a label that's very different. Their label is CD105 or CD90. Here we're in CD4 and CD8. So
But they're not just T cells.
they're not T cells. They're mesenchymal stem cells. I was just giving you an example of how all cells in the body have these labels on them.
Okay.
Yeah. So one of the ways that we know that they're mesenchymal stem cells is because we can stain. You can actually use an antibody to target these labels, and then you know that that's what it is. That's how you identify it. Okay. Other questions so far? Yeah. Yeah. So just like this helper T cell is displaying this label. It's like a name tag. It's like, hello, my name is Rob, right? It's like it's a name tag. It's one of the ways that cells actually interact with other cells. Is we'll see that here in a minute. Is they identify this name tag and they know what to do based upon these markers on the surface. I mean, did you ever wonder, like, when two cells get together, like, how do they know what's going on? How do they even know that they're cells from the same organism? Well, there's a label for that, and we'll get to it, okay? These are the Mhc complexes. We're going to talk about that in a second. So all of your cells have these labels on them that tell your body what kind of cell they are and what they can and cannot do, okay? Other questions so far? Can like be a spy? Can a
Yeah, can it have say a mutation in its DNA to create a protein that pops up on its membrane and uses it to infiltrate specific parts that
Yeah, so I mean, some of the more clever ones do that. So for example, when we look at HIV, it kind of does that. It's an RNA virus, and it uses a caspid to get in, and it's a little bit like a Trojan horse type of thing. Yeah. So I mean, in virology, microbiology, I mean, these bugs are really clever as how they circumvent the immune system, right? So, I mean, if you think back at the roadmap that we just had, that's an awful lot of defense mechanisms to keep things out. But we get sick, right? 48 states have the flu virus right now. That's a really bad one this year, right? And so the mutations are happening, right? The coronavirus, over 800, I think, casualties now from the coronavirus. I mean, it's getting pretty bad. So, infectious agents are clever of how they're getting around it. But fortunately, we do adapt as well. And so let's talk about this. So, I mean, we've spent now, I don't know, good 10, 12 minutes, and we're on the title slide, okay? I haven't even, gotten to slide 1. This is, the title slide. But it's really important for us to have that background, because did you guys find that helpful just to see where everything fits in? It might help explain some of the reading that you did, because it's super confusing. I would say this section is one of the most challenging sections for students, and that's why we kind of slow things down intentionally on this particular part. But we're going to dive right in fast. Here we go. Hang on. You ready? Hopefully you're buckled in, because we're going to get going. So third line of defense, like what we had on the board, also referred to as specific immunity. We've got two main types that we characterized in our outline. We've got CD4 and CD8 cellular mediated, but we also have antibody mediated with the production of B cells that crank out antibodies. Okay? So this is all in line with the outline that we had up on the screen. In prior lecture halls, I could have the whiteboard up at the same time as the screen. This one's a little bit more complicated, so kind of had to do that first, and now we move on to this. But hopefully you wrote that down. You can refer to it. I can put the screen back up if we need to look at certain things later in the lecture. But what I want to walk you through right now is I want to walk you through humoral, right, immunity, is another name for our antibody-mediated response. And then cellular immunity is the name that we give for cell-mediated, or T cells. So you can say this column is basically B cell activity. This column is basically T cell activity. Underneath the category of our adaptive or our specific immune system. So we got a bacteria that comes in. This bacteria, right, that's invading is seen. It's actually seen by a B cell, and the B cell secretes an antibody. So these little blue Y-looking structures, these are monomers, and they find the bacteria and they bind them up. And they bind them up, and a number of different things can happen when they bind. They bind up. They could, for example, be phagocytosed by a macrophage. If they're phagocytosed by a macrophage, then you know that that bacteria could be killed by that macrophage using what mechanism? That could be on the exam. Oh, yes, that one. Dang it. How does a bacteria die? How does a macrophage kill bacteria? Could be enzymes. What if they're resilient? Phagocytosis just means it internalizes it. How does it destroy it? This one says it's destroyed. Here it's phagocytosed. Here it's destroyed. Oxygen. What happens with oxygen? What do? Chokes it with oxygen. Really? You're close. Someone help Max out. He's getting super close. So you guys play that game, getting warmer, cold, right? Warm, right? Yes, sir. Reactive oxygen species. Well done. Remember, at the membrane of the phagolysosome, we utilize oxygen and we create a free radical, but it's contained in a vesicle inside of the macrophage, right? And now the bacteria was in it. So the macrophage engulfs it and becomes a phagosome. It merges with the lysosome. Now you have a phagolysosome. The lysosome had the enzyme. Max was halfway right. And then the phagosome has the bacteria. They come together right? And then you generate reactive oxygen species, like detonating a bomb inside of a controlled container, right? Okay, good. So that's one fate of the bacteria. We'll look at some other things that can happen here within this process. So like this one characterizes it right here. So this one here, these blue antibodies bind and they neutralize it. So one is they could bind up the bacteria and just neutralize it if it's not that aggressive. What's that? They neutralize it. So one way that they can neutralize it is they bind up all of the binding sites and they render it like immobile. So it just sort of like gets bogged down. This red antibody here is the one that got phagocytosed. There's another example where they could actually do lysis of the bacteria, and that goes back to triggering complement. You guys remember a complement with the Mhc complex? Okay. All right, so that's over on the humoral side. On the cellular side, we've got a microbe, right, that's intracellular. We've got an antigen-presenting cell that actually is showing to the helper T cell, which is a Cd4 positive T cell. The antigen-presenting cell is saying, hey, look at what I found. And the helper T cell says, ooh, that's interesting and looks dangerous. I actually better blow the whistle. So it blows the whistle. That's releasing cytokines. That triggers more T cells to be recruited and activated. And these that are cytotoxic T cells, which are CD8 positive, that are activated go and they actually go attack the infected cell. So this would be cellular immunity because cells are involved. Humoral immunity because the antibodies float through the bloodstream. Okay? So locations. Humoral antibody happens in the blood. That's why it's called humoral. Humors, back in the original Roman times when they were describing a lot of anatomy and physiology, they talked about a number of different humors that were made up of. So blood was one of the humors. Okay, urine was one of the humors. Okay, not like funny humor, like humor like a fluid medium. So humoral is referring to the blood medium. In cellular, this could circulate in the bloodstream or it could circulate in or migrate through tissues because they're cells. So location, you could write on the screen or write on your page. This is blood-based. This is blood or tissues. Okay. All right. I introduced the antigen-presenting cell right here, which we abbreviate APC, antigen-presenting cell. Its role in life is to showcase stuff that it finds, antigens that it finds in the body. And it's going to walk up to things and try to figure out, are you part of our host or are you foreign? That's like the first question it's going to ask. Okay, and then if it's foreign, it wants to understand, like, how bad is this? Like, how much backup do I need to call? If you guys think of the storyline for the immune system as like a defense mechanism, like military style or like police style, that will help you understand like how this all interacts. Because that's a lot of the readings that you're reading. If you think of it in that context, like you're trying to defend a nation, you're trying to defend a city, that's sort of the storyline that the body uses. Okay. So some unique characteristics. And now I might actually need this guy, Richard. We're going to do this. Let's go here. See if we can get this on the... Okay, that might actually work. Okay. Unless it's better. Okay, we'll try that. So, now what I want to focus in on is some of the unique characteristics of these cells and how they interact with each other. Okay, we're like on slide 3. Okay, it's like 40 minutes and we're on slide 3. So up top here, we've got an antigen-presenting cell. And down low, we've got a cell that's displaying Cd4. So what kind of cell is down here? Helper T cell, very good. Okay, so let's draw this. So we've got a cell down here, and then we have a cell up top. Okay? This guy's got Cd4, so it's a helper T. Well done. This guy is an antigen-presenting cell, an APC, and it's actually displaying a molecule that we refer to as major histocompatibility complex 2. This is called Mhc 2. Okay? So do you see, here's a phospholipid bilayer. So this is cell number 1. Here's a phospholipid bilayer. That's cell number 2. Do you guys understand the architecture? So notice, this is much more complex than my drawing, okay? First thing you should notice. Fair. But notice that the CD4 interacts with the MHC2. And the MHC2 is holding something of interest. It's holding the antigen. That's that yellow structure. So I really should draw this a little closer together. You're like, dang it, Keller, I just put it in ink. Sorry. And this guy actually has some sort of antigen that it's displaying. So it's displaying this antigen to the helper T. Now, the analogy that I like to use, I don't know, back when I was growing up, it was the Cold War era. So we were always worried about, nuclear warfare with Russia, exactly. All right, so as little boys, you'd play like with your G.I. Joe figures, and always America versus Russia. That was always the game, right? Well, back in those days, we'd always see these movies, and when you wanted to do a launch sequence for a nuclear strike, there wasn't just like a red button that said fire the nuclear warhead, right? What was the setup? There were 2 keys, right? So like one general had a key, and then another general had a key, and they would go to the console, they'd put their keys in, and they'd go, all right, 1, 2, 3, turn, right? And then the button would light up, right? Do you guys remember what I'm talking about? That's kind of how I visualize this. I tell you that goofy story because I think it helps. These two are interacting, and we want to make sure that that antigen that's being presented is actually a legit foreign invader. You don't want these two, the CD4 and the MHC2, to find each other and just kind of brush up against each other, and then you activate the helper T. And the helper T is going to blow the whistle and recruit cytotoxic T cells. So you have 2 signal events. And these 2 signal events equal what we call co-stimulation. So you read about co-stimulation in your text. We're going to talk about what that looks like right now. So signal 1 is this event right here. So you've got an alpha and beta chain on the MHC2 complex. It binds to the CD4 portion, and then there's this TCR heterodimer that's stuck in the membrane that has all these different subunits. This whole thing is one protein, okay? And it's got a number of different subunits. It also has an alpha and beta chain. But the take-home message that I really care about is the peptide antigen binds to this entire complex and initiates signal number 1. So right here on our simplified drawing, this right here is signal 1, okay? Aha, but that's not enough, because that could accidentally happen, and that could be a bad thing. So we have this signal number 2, and that involves 2 different molecules. Here on this helper T cell, it's the CD28, right? And then on, let's go MHC here, 2, get rid of this. Over here, it's this molecule, and there's a couple of different types that it could be. It could be CD80, or it could be 86. And when these 2 interact, that's signal 2. And it takes signal 1 and signal 2 happening at the same time to trigger activation of that helper T cell. And when you put these 2 together, this is co-stimulation. Okay. Does that make sense? That's correct. Yep. So signal 1 is this interaction of the legitimate peptide antigen binding to all of the TCR heterodimer that's interacted with the CD4 plus the MHC complex. When this happens and signal 1 is triggered, okay, and it's a legit signal, meaning this is an antigen and all these pieces are in play, it triggers colocalization of the CD28. Okay, the CD28s come into the nearby vicinity and they're finding the CD80. So if this happens on accident and they all get together and you don't get colocalization of CD28, then you don't get activation of the helper T cell. Okay, so now let me ask this question. This is just helper T cells with Mhc complex 2. That's all we've covered so far. How many possible things could you identify that could go wrong if we have a disease state? One? One thing? It's pretty simple, not that complex. Or are there multiple moving parts here? And does it surprise you that we have a breakdown and we have immune issues in our patient population? Okay, we've literally just covered helper T cells interacting with antigen presenting cells, APC, that has an MHC2. That's it so far. And you're already seeing, wow, there's half a dozen to maybe a dozen different things I could identify just theoretically that could go sideways and we'd have a problem. We'd have helper T cells that don't activate, or we could have helper T cells that are active when they shouldn't be active. Yes, sir.
So where do these like CD28s and CD8s, where do they come from? Are they like, as soon as the cell gets sick, they present this, or?
So they're there, and so they'll colocalize means that they move in the membrane. Remember, the membrane's fluid. You guys remember this? The phospholipid bilayer is a fluid membrane. What does that mean? So the analogy that I would give is when I taught basic bio classes, how many of you grew up with a pool? Okay, imagine styrofoam balls that are about this big and put white styrofoam balls to cover the pool. Then take out maybe 10 of them and color them blue, and take out 20 of them and color them yellow. So those blue and yellow balls are sitting in this fluid membrane, right? And the whole surface is all white with like 20 yellow spheres and 10 blue spheres. And those yellow and those blue spheres are different proteins that are stuck in the membrane. Well, you could grab a yellow sphere, you could go underwater, kind of be funny, be inside the cell, reach up, grab a yellow sphere, and drag it from the shallow end to the deep end, right? And that's how you would move a transmembrane protein. So they exist, but they move into an area that they're supposed to be when signal 1 happens. Does that make sense? Yeah. So it's a legit question. The other thing that can happen is you can trigger the nucleus to say upregulate the production of CD28. Like I need more of them. That takes a little bit more time, right? To make a new yellow sphere or a new blue sphere. It'd be easier just to grab it and move it through the membrane.
But they're always there.
They're there.
If it comes in contact with something it's not supposed to, it's going to move it.
That's correct.
So it's going to be more likely.
Yep.
To
Yep. Yep.
So this antigen presenting cell, is this one, like is the CD80 or 86, is that only specific to that cell, like the MHC2, or are all like antigen presenting cells, do they all have either CD80 or 86?
All of them are going to have either 80 or 86. That's correct.
One's just specific to the MHC2 one?
This one, well, so there are different types of antigen presenting cells. So, for example, macrophages are an antigen presenting cell. Dendritic cells are an antigen presenting cell. And those two have different CD nomenclatures. Other than that, I don't know which ones have other, but there are other types of plasma cells, like B cells, can be antigen-presenting cells. Okay? But all antigen-presenting cells are going to have that fundamental architecture. Okay. All right, let's look over at the bottom right. So the bottom right picture. This is antibodies. This co-stimulation event occurs with antibodies as well, not just with T-cell interaction. But with antibodies, we've got, here's our monomer, right, IgM, and it's bound to an antigen. The co-stimulate, and that's when the antigen binds to the antibody, that initiates signal number 1. Signal number 2 comes from a CD21 binding in the presence of complement. Okay? So we've got a simplified version. Let's talk about this simplified version here. We've got an antibody. We've got an antigen. Oops. That's signal 1. And then signal 2 is initiated by a separate protein. This one is CD21. And it interacts and binds to create that secondary signal with complement. Now remind me, what is complement? Where does it come from? What makes complement? Well, this is a co-stimulation event right here. This is signal 2. Okay, so together that's co-stim. And this is for antibodies. This is co-stimulation for T cells. So B cells and T cells. I'm asking a question which is kind of a review question. What is complement? Tell me about it. Where does it come from? We've already learned about it. Where did we learn about it? Inflammation. We learned about it in the inflammatory lecture. What do we know about it? Back here. 1 second, Max. Back here. It's a protein. It's made by the liver. It's used to either complement the inflammatory response, or in some cases, like when it forms a Mhc complex, it'll go directly lyse and attack bacteria. That's correct. Okay. So now I want to ask a question, and I think I need to put the screen up for this one so you guys can kind of see the whole thing. Are these different parts of the immune system mutually exclusive? Do your first, second, and third line of defense, are they mutually exclusive? Or is there some overlap? If there's overlap, give me an example of an overlap. Correct. Yep. How about complement protein? That's true. That's absolutely true, Max. That's one. There's many. How about complement protein? Did we just not learn about how complement triggers signal 2 in B-cell mediated adaptive immune system? So these things actually overlap and are helping each other. There are not hard and fast lines. You guys have to remember, the body doesn't say, oh, I'm part of the first line of defense, man. I don't go there. Like, I'm a macrophage. I'm the second line of defense. This is all you. This is like third line of defense material. No, you have to remember, in the human body, everything is very fluid. And we put labels and categories on things because it's easier for us to understand it. But everything is working in concert, right? I mean, the heart and the lungs are different organs, and we study them in different systems, but they are totally intertwined, okay? So we're presenting it very, very rigid and very, very linear, and I just want to make sure that you guys can appreciate that there are a lot of overlaps that take place in the body. Okay. Lymphocytes. We've covered some of this material, but now we're going to dive in a little bit deeper. Where do they come from? So they come from the bone marrow. These lymphocytes come from the bone marrow. And in the bone marrow, from lymphatic tissue, which is what's in the bone marrow, we're going to see a number of different things arise. And so on this slide, what I want to characterize is I want to characterize this concept of these pro-T cells. If we've got primary lymphoid organs like bone marrow and thymus, bone marrow, we said earlier, is where B cells actually mature. And we use the language of become immunocompetent. They become educated. They learn, hey, if this is part of the host, don't destroy it. If this is not part of the host and it's foreign, let's get rid of it. That's a very important decision. And so what we say is, we say this thing is this self or is this non-self? So if it's self, do you want to kill it? No. If it's non-self, typically, what? You kill it. So these bone marrow cells can either become mature B cells and make antibodies, and we'll talk about those in a minute. Now we're going to talk about them going to the thymus and them becoming immunocompetent. T cells. If they are self-reactive, that means they're going to find an antigen like on your pancreas and say, destroy the pancreas. Yeah, you guys look at me like, holy crap, that sounds aggressive. Yeah, well, that's what happens with diabetes type 1. Right? So if they're self-reactive, then they have been programmed to go react with tissue that is your own. Well, in the thymus, we have this process known as central tolerance, where we're going to say, we're not going to allow that. We're going to screen for any self-reactive clone. And if it's self-reactive, we're going to trigger it for apoptosis. Ah, I know that word. Apoptosis is what? Programmed cell death, right? So we're going to say, no, I'm not going to let you out of the thymus. You're not going any further. We're going to recycle all your parts. We're going to try this again, soldier. But no, you cannot be self-reactive. So then, theoretically, the thymus will only release non-self-reactive clones out into the system, and you have the ability for the induction of immunity. So it's non-self-reactive. It goes out here, it finds a non-self peptide, and the stuff of stimulation number one, or signal number 1, signal number 2, co-stimulation, go ahead and stimulate a T cell response and go kill the non-self infected cell, right? If it's infected with a non-self protein, then we're going to kill it. And that's what we just covered. So that's how it's supposed to work. Every now and then, central tolerance doesn't work. And you get something that leaks out of the thymus, and it is self-reactive. And then we have a backup mechanism with T cells, and we call it peripheral tolerance. And that means it's not central, it's not in the thymus, it's out in the periphery. And we have 3 mechanistic pathways for peripheral tolerance. Number 1, anergy. Number 2, suppression. Number 3, apoptosis again. It's coming back. If you wind back and you look at the old lectures when we talked about necrosis and apoptosis, I told you there were 2 examples, and one was between our digits of programmed cell death. And I alluded to the second example was in immunity, right? The immune system. And this is it. So we use apoptosis. We stopped using apoptosis because our hands are already formed, and our toes are already formed. But we use apoptosis to get rid of self-reactive clones all the time. In fact, if you look at the numbers, the body errs on the side of let's be creative and make all these different variations of clones. And we know if we're very creative about moving things around and trying to anticipate all the different types of antigens that we're going to see, inevitably we're going to react with our own tissue. Does that make sense? And so 90 to 95% of the T cells that your body makes are self-reactive, and you recycle them. So that should tell you we spend a lot of biological energy. It is very expensive to run this system because you're recycling through apoptosis, either central or out in the periphery. You're recycling more cells. Far more cells are being triggered to try again because they're self-reactive. But we want to try to anticipate. That's why we have this immune system. We want to try to anticipate what are we going to come across. You with me? Question? Why do we make so many of the T cells? Well, so what I'm trying to explain is, if we're sitting down at a table and you're trying to draw out all the different variations of how you can make a T cell respond to different proteins, right? You could say, okay, well, don't make it respond to anything that's like me, and we stay away from all these things, but then if a virus or a bacteria is clever enough and tries to mimic you, then it gets through. And so we spend a lot of energy, biological energy, trying to come up with all these permutations, and then if it becomes self-reactive, we get rid of it. And so we spend that energy so that we can have a very widely diverse adaptive immune system. Versus the conservative way to do it would be, don't try to predict, just make classic this, this, and this, but then more bugs would come through. Okay? These three processes in peripheral tolerance, outside of the thymus, anergy. If a self-reactive peptide binds to the Mhc complex, but it lacks co-stimulation, it signals the cell for anergy. That's a word that just means it doesn't do anything. It kind of just gets stuck in this phase of non-reactive. It doesn't go anywhere. It doesn't die. It just doesn't do anything. That's if you get no co-stimulation event. Here, let's go to this one over here. If you've got a self-reactive peptide that binds and you get this co-stimulation, but this is self-reactive through this Mhc complex, this is when you trigger apoptosis. You say, oh, try again. Okay? Try again. This one is the category that's far less known, and there's a lot of, most of the new research in immunology. This is not my field of expertise, but I try to stay up on it as much as I can, and a lot of the new research that others are doing in the field is with these regulatory T cells, or suppressor T cells is another name for them. So these are a cell that comes in here and literally is like the negotiator. And so even this is a self-peptide with co-stimulation, but because it's self-peptide, it must signal this regulatory T cell to come over, or a suppressor T cell is another name for it, and kind of calms this whole situation down and says, don't activate. And that's an outside cell that's doing it. They're out in the periphery, and they're kind of like the regulators or the oversight that's taking place on the immune system. So in the immune system, for the T cells, we have central tolerance in the thymus, peripheral tolerance out in the periphery, and we got three different mechanisms of peripheral tolerance to keep away self-reactive clones. This is a picture of the thymus right here, location-wise, like I was telling you about. And, just a couple of factoids about this. So it's a pinkish gray colored tissue. It has lobes, so it's lobulated. It's about 5 centimeters in length when you're born, about 4 centimeters in width, and about 6 millimeters in thickness. So it's not that big. But it grows in childhood, and it reaches a maximum weight at puberty of about 20 to 37 grams. But then by the age of 75, it's about 6 grams. That's it. It shrinks down. So these primary lymphoid tissues that we talked about, bone marrow and thymus, there's secondary lymphoid tissues that things go to in order to circulate through the body. And in these secondary lymphoid tissues, this is where we're going to get activation that takes place, because that's where it's going to experience things. So secondary lymphoid tissues, for example, how many of you have had your tonsils removed? Wow, that's quite a few. Let me see again. Just kind of curious. Okay. How many of you, like, if you get sick, like the sides of your neck are really sore, or maybe behind your ears? Some people. How about like your armpit? Have you ever had like a tender armpit? You're putting your deodorant on, and you're like, oh man, that's kind of tender, and then the other one's not. It's kind of weird. Okay. This one's a little bit more awkward, but how about like in your groin? A little tender in the groin. So those are places where the lymphatic circulation goes to lymph nodes. You have lymph nodes throughout the body. So this was in 202 when you were looking at the circulatory system and the lymphatic system. That's these lymphocytes that circulate through that lymphatic system. That's the roadways by which they are trying to come in contact with viruses and bacteria. Okay? Out in the periphery. So how are they activated? Well, we talked about this slide at length. So hopefully this makes a lot of sense. Hopefully this is understandable. Hopefully there isn't anything here that doesn't make your notes kind of go together on activation that requires co-stimulation, whether it's T cells, co-stimulation, whether it's B cells, okay? And so these tissues, these cells are going to operate through what we call toll-like or T cell receptors or B cell receptors. That's the broad category of these systems that we've been talking about. T cell receptor activity, B cell receptor activity. And it all kind of pivots down to an antigen. So you need an antigen to make this whole thing go, but it's not the only thing that you need, and now we know that, right? So when we get this thing to go, and we're circulating around the body, what does it look like? Well, this is a diagram of a lymph tissue, or a lymph gland, or a lymph node. And you've got little valves, one-way valves, if you remember the lymphatic circulation, kind of like the venous circulation. And so we've got a lot of things that can come in, okay? And this is a fluid system that's flowing, okay? And it's like a filter. Those lymph tissues, like your tonsils, your lymph nodes in your armpit, axillary lymph nodes, the ones in your neck, the ones that run all the way down, and there's kind of locations at every joint in your lower extremities. That's why sometimes in your groin, that's why when you get sick, that's a little tender, is because there's probably some infection in that lymph node. So in these, we've got what are called antigen-presenting cells. We talked about antigen-presenting cells. Well, these antigen-presenting cells could either be B cells, they could be macrophages, which is the one that we've heard about the most, or they could be dendritic cells. Now, dendritic cells are not related to the nervous system. They are not nervous tissue, dendritic cells. They look like dendrites because they have kind of like that branchy appearance, but they're not nervous tissue. Dendritic cells live within the dermis, in the skin, which should make sense because their location is close to the external environment. So if something gets through the skin, through the barrier, those dendritic cells are there. You with me? So B cells, macrophages, dendritic cells, 3 types of antigen-presenting cells. At times, plasma cells, which are a type of B cell, can also be an antigen-presenting cell. What are some cells that are not antigen-presenting cells? Neutrophils are not. How about fibroblasts? No, they're not antigen-presenting cells. Okay. These are specialized cells that present the antigen. These cells can present either to CD4 or CD8, so they can go helper T or they can go cytotoxic T. And they're going to do this in the lymph node or in lymphatics. So let's walk through this diagram. Here's a dendritic cell with an antigen. comes into the lymph node. It can go either CD8 pathway to the right or a CD4 pathway to the left. Let's follow the CD4 pathway first, and then we'll get over to the right. CD4, which is a helper T cell, right? This antigen-presenting cell presents to the helper T cell. That was what we spent kind of our first mechanistic conversation on, right? That's what we drew on the board. And when it presents here and we've got co-stimulation, then we get these helper T cells that are activated and recruited. They move out into the circulation. They go and find in the tissue, right, a phagocyte out there that's been ingested a microbe. It's displayed it, and then they trigger cytokine release, and either the macrophage is going to kill it, or they're going to bring in more inflammatory cells out of the circulation by recruiting these macrophages out, which is part of the immune system, but it's our second line of defense known as inflammation, right? And how is it going to, with cytokine secretion, how is it going to get this cell out of here? How is it going to get that cell out of here? So angiogenesis is a component of it, but how do you recruit this monocyte out here to become a tissue macrophage? Pancake, yeah. I heard rolling. So margination would be first, right? Is blood clotting important for margination? Yes. It helps control blood clotting, helps slow down the blood flow. Margination means those cells move to the edge, those monocytes move to the edge. Then what molecule is upregulated to make that endothelial cell to that monocyte interaction a little sticky? Not yet. Selectins, are they a weak affinity or a high affinity? They're weak affinity, and they're in a blood flow environment, so what happens? Do they stay or do they get released? They get released, and that triggers what? Rolling. As they roll, that upregulates expression of what molecule? Integrins. Integrins bind weak or strong? Strong. So now the monocyte is stuck. We call that tethered or adhered. And then it migrates through. We call that diapedesis or transmigration. Okay? And that's how you get this here. You guys with me? So this box that says inflammation, that's what that really means, is what I just said. And you're like, well, that's not fair. It says one word. I said, I know, but that's what you just learned, is all of those steps on getting that monocyte out here. And that's what this T cell is triggering. So now you just had an example of your third line of defense using your second line of defense to battle. Make sense? All right, let's back up to our CD8 positive T cell. Backing all the way to the start, the dendritic cell has an antigen. It shows it to a CD8 positive T cell, which is also known as cytotoxic T cell. And there's a blow up here of this mechanism that's happening here before it gets activated. So let's go take a look at this. So this interaction of cytotoxic T cell and a CD8, a cytotoxic T cell with the antigen presenting cell, you got your APC, you've got your cytotoxic T cell that's naive, is the definition. Naive means it hasn't yet been activated. So cytotoxic T cells have a unique type of co-stimulation that's a little different than what we learned right here. This was not cytotoxic T cell. What was this one? Helper T cell. Now we're going to talk about cytotoxic T cell co-stimulation. It involves antigen binding, and then what happens is you've got interleukin, which is a cytokine that gets released, and it binds to itself. It binds to the cell that released it, which is the cytotoxic T cell. That binds to an interleukin receptor, which is now an autocrine response. And that is what activates cytotoxic T cells. So the co-stimulation event for cytotoxic T cells is interleukin-2 binding back to the same cell that made it after it interacted with an antigen-presenting cell. Does that make sense? Still co-stimulation, but it doesn't involve the CD80 and the CD28. It involves interleukin-2. Okay? Now it makes two populations of cells. It makes effector cells and memory cells. These effector cells move out into the population, and they can trigger a response against a contaminated or infected host cell, one of us, and it just signals it for death. It kills it. Not in an apoptosis, it actually kills, it destroys. They're cytotoxic T cells. They literally go out and kill the cell that's infected with the microbe. Okay. Crazy, huh? Well, this is happening. It's bad if this happens to a tissue that is not infected. True.
But I'm saying even if it was infected, what we talked about with the necrotic tissue, that when it explodes and just
Yeah. It is bad, so the next cell that's going to show up after this is going to be what cell type to clean up the mess? Macrophages. Okay. All right. Last slide for today, and this is where we're going to pick it up. Two quick slides. This one and then the next one's really fast. We already talked about the next one. Antigen-presenting cells. We talked about different types of antigen-presenting cells. This is a dendritic cell. This is a scanning electron micrograph of a dendritic cell right here. And this is a lymphocyte. So I want you guys to appreciate this close interaction. I mean, they literally get right up in each other's space. There is no personal space. Okay? And here is an MHC2, right, with a effector T cell. And you've got your T cell receptor and your CD4. This diagram is just like what we drew earlier. It's just a different picture out of a different book. So co-stimulation is happening here. Over here, this is a macrophage, right, showing the shape of a macrophage. Here's bacterium coming in, and this is the antigen being displayed on an MHC2, and then it goes to the surface just like we already drew. I just want you to see different types of pictures of the same thing. You guys with me? And then last, these are 3D representations of these major histocompatibility complexes 1 and 2. What they are is they are what makes you you. So all nucleated cells have an MHC 1 or 2 on them that's like your name tag, like, hey, I am Rob. Okay? And this is the point, or this is the problem with transplants. When you have a transplant rejection, or you have to have a match. And some students want to ask, like, well, is that the same thing with blood? No, blood is not nucleated any longer. Erythrocytes were nucleated when they first were born, and then they expel their nucleus. You still have blood matching, type matching that has to happen, but it's not MHC. It's different types of antigens. Antigen A, antigen B, or antigen AB, and then you have the rhesus antigen. But all nucleated cells, like if you take a kidney out of one patient and you put it in another, it doesn't take because of the Mhc complexes. But we use these to display antigens, okay? And I just wanted you to see the architecture. The MHC1 presents to CD8 positive. And what are CD8 positive cells? Cytotoxic T cells. The MHC2 presents to CD4. And CD4 positive cells are what? Helper T cells. There's no great way to remember this. Just memorize that part. All right, I'll see you guys on Wednesday.